Hello, this is Lecture 2 in Social Neuroscience, and today we're going to be talking about methods that we use in social neuroscience. The specific methods that we're going to focus on are ones that really are used with humans. And I don't want to give you the impression that we're not interested in non-human animals. In fact, social neuroscience does investigate everything you can possibly imagine, like ants and goldfish and chimpanzees. But to keep our lectures sort of in bounds here so I can get something done in the 26 lectures that I have, I'm going to focus today on methods that we commonly use with humans. And so the things that I'll be covering today include peripheral measures, measures of hormones, behavior and self-reports, behavioral genetics, and then we'll go through and talk about how you can study the brain more specifically. So just to think about this a little bit, we do have 26 lectures, and I didn't really talk about them in the last lecture, but if you look across our 26 lectures, what's going to happen is these first 12 are really going to be covering specific levels of analysis. So we're going to be going through the different levels that I talked about in the last lecture. And as such, you can kind of see that a lot of these topics have methods that would go with that particular level of analysis. So that's what I'm covering today, are these methods that could be used across the nervous system, the endocrine system, genetics, and so on. And so that's just, again, to give you an overview. After we get past that, those first 12 lectures, the remaining 14 then are going to focus more on specific topics that have to do with social neuroscience. All right, let's move on then to peripheral measures. So peripheral measures, by what I mean by this, these are kind of what was traditionally called psychophysiological measures, but these would be measures that you attach to a person's body. They're not generally invasive, although sometimes they can be. They can break the skin. But most of the time, these are just electrodes that sit outside the body, below the head, and measure activity from the body in various ways. And so what I've done is I've grouped these different measures, the main ones I'm going to talk about in this lecture, based on where they're recorded from. So for instance, we can start with the skin. And yes, there are electrodes that you can put on your skin, particularly on your fingertips or the palm of your hand, that measure what's called electrodermal activity, or EDA. This is also known as GSR, or the galvanic skin reflex. That's an old term for it. Also, you could call it skin conductance. And what you're doing is you're basically sending electrical activity or a voltage across the skin, across between two electrodes, and you're measuring the sweat gland activity underneath. The sweat glands sort of act like resistors, and you can see how much the person's sweating or how activated their sweat gland activity is between those two electrodes. And the sweatier they are, the more the signal is going to be conducted. And so that's what's done here. You can see we got a couple of electrodes here on the person's fingertips, and then what you see on the screen is sort of what electrodermal activity looks like. It's really slow. It goes over several seconds and minutes that you see these kinds of changes. It's not a very quick response. And so you can kind of use this if you're kind of measuring like a person's overall level of arousal or excitability. Another peripheral measure would be ones that measure heart or cardiovascular activity, and there are actually quite a few under this category. For instance, we have the old common electrocardiogram where you put electrodes on the chest or on the arms to measure your heart activity. You can see the EKG signal. There's a very specific form of your heart cardiac cycle that you can see. You can measure how fast the heart is beating, what the heart rate is. But you could also include under this category things like measures of blood pressure. You could also measure blood flow out in the periphery, like how much blood is out at your fingertips. You could even talk about respiration as being one of these kind of cardiovascular measures. One of the ways that cardiovascular activity is commonly recorded today is using what's called impedance cardiography. And this person that you see in the photo has some electrodes on there for impedance cardiography. It's not as big as it looks here. But basically what you're doing is you're measuring voltages that go from like up in the upper chest down to the lower abdomen. And what happens is this electroactivity goes along major arteries, and you can use that to measure lots of features about the cardiac cycle. So you could measure, again, just the regular ECG like you see in the top of this figure, but you can also look at things like how much blood flow there is, how much time it takes before the blood fills in the chamber of heart and gets pumped out, and what that time is. So these are all very specific variables that you get there down at the bottom that let you really get into looking at cardiac activity. And this is actually used a lot in social neuroscience. Impedance cardiography has been looked at in terms of sort of like the stresses that might happen when you're interacting with strangers. It could be used as a measure of inequity. So when you are not being treated fairly and someone else has got something that you wanted, you can see studies out there that use impedance cardiography. All right, the next class of peripheral measures are ones that measure muscle activity, and this would be the electromyogram. Now, the electromyogram can be recorded by sticking needles right into muscle tissue and measuring the muscle as it fires. They do this, for example, in human movement studies and in physiology. In psychology and in neuroscience, we tend to use the electromyogram on the surface, and we put electrodes on the skin and measure the muscle activity that's just below that place where the electrodes are. I have specifically in my research used EMG to measure facial muscles, facial muscle activity. And you can see here what's going on is that the researcher is sticking some electrodes above this woman's eyebrow and on her cheek. And what they're doing is those electrodes are measuring muscles that you use for different facial expressions. So for example, if you look at this, this is actually meant to be a representation of all the facial muscles that you use to make different facial expressions. And it's kind of like if you took your face and just took it off and looked at the inside of your face, you'd see that there are all these muscles just underneath the skin that are attached to the skin and to the bones that help you push around your face to make different facial expressions. So these are just used mainly for facial expressions. They're not really used for talking or chewing. They're used to help us convey signals to other people. And as I said, with all those different muscles there, the two that we tend to look a lot at in EMG, in facial EMG, is the corrugator, which is there above your eyebrow, and you use that to furrow your brows to make negative expressions. And then you can also measure this muscle that pulls the lip corners back to make a smile. And so this is these are two common sites that you find in facial EMG. Here's again another participant all hooked up. You can see she's got the electrodes on her cheek. She's got the two up above her eyebrow. There are also two there below her eye. What those are doing is they're measuring a muscle called the orbicularis oculi, which is a big muscle that goes around your eye. You use it when you blink, and so you can actually measure how much a person blinks using that. But you can also use it to measure the muscle that you use when you are smiling really happily and you kind of crinkle up your eyes a little bit. The orbicularis oculi would become more active in really positive states. So the way that facial EMG has been used by social neuroscientists is to measure it as a measure of affective valence, basically how positive or negative a person is feeling. So you can see over there on the left, the corrugator is that brow muscle, the one you use to furrow your eyebrows. And you can see when a person looks at moderately negative stimuli, like pictures of a dead animal or garbage or something, versus something that's moderately positive, like an attractive model, you can see that the corrugator activity is the greatest for these moderately negative and the least for moderately positive. On the other hand, we have zygomaticus major, the one that we used when we pull our lip corners back, and you can see that when you look at moderately positive stimuli, you smile quite a bit, and when you look at moderately negative, you smile just a little bit, so it goes back down. So these two muscles sort of work in reverse, and in fact, it's kind of hard to both furrow your brows and smile at the same time. If you try that right now, it feels weird because usually what happens is when the zygomat goes up, the corrugator becomes more relaxed, and vice versa. So this has been used in lots of different ways over the years. It was first really developed back in the 70s and further developed by Cacioppo and his colleagues in the 80s. I've used it in my research to measure racial prejudice. And we found, for example, when you look at somebody who's in a different racial group from you, you might actually frown more, show more corrugator activity and smile less, and do just the opposite for people who are in your same racial group. All right, another class of peripheral measures would be the electrooculogram, or the EOG. The EOG is recorded by putting electrodes on above and below your eyes, to the left and the outer parts of your eyes. And what happens is the eyeball is sort of like a big battery. It's got a positive and a negative charge, a dipole in the eyeball. And as your eye moves around in your skull, it actually changes the orientation of the dipole. And with those electrodes, you can measure whether the person's looking up or down or left or right. And so EOG has been used to just measure eye movements, like when people are reading, when they are looking at different kinds of stimuli. These days, you don't see EOG being used as much as it used to. It's used more as a way to measure artifacts, that is, a way to measure that someone isn't blinking when you don't want them to be blinking, that kind of thing. Instead, people use eye tracking these days to really look at eye movements, and we'll talk about eye tracking a little bit later. And then another kind of peripheral measure would be to measure stomach activity. Yes, you can actually measure how the stomach is digesting food through what's called an electrogastrogram. So again, you put some electrodes on the surface of the skin, down over your belly, near your stomach. And what happens is, as your stomach is digesting food, it very slowly does this. It creates these EGG waves that are very, very slow, 3 times per minute. They're cycling through. And then they change when you're more active or you're more stressed versus you're more relaxed. And that kind of makes sense because, of course, things like stress are going to affect how your food is digested. And so these muscles in your stomach walls, as they kind of very slowly munch up the food there, they're changing their activity of their waves. And then you can use those waves then as a way to kind of measure how much stress the person is or how they're emotionally responding to a particular stimulus in their stomach. And then finally, one of the newer measures here on the list is what I've put down for peripheral nerves. There's something called microneurography, or MNG. This is a technique that's just been around in the last couple decades. It was originally developed in Sweden, and it involves inserting really super skinny, tiny little electrodes or wires into a person's skin, right into their nerve. And you do this in peripheral nerves, like in your arms, your legs. Just recently, I saw on Twitter a lab that was doing this. You can see here, she says, nothing to see here, just a live recording of action potentials from single neurons in an awake human. And the guy that's sitting there in the chair is actually having his nerve recorded in his arm while he's awake, and everything's fine. He doesn't feel any pain. And then they can present stimuli to him and see how that goes. Now here actually is that lab. They have a little video that I found, and you can watch it here. So what this lab is actually doing is they're using MNG to measure what are called C fiber neurons in your arm that are sensitive normally to pain, but there's actually some little sub tracks of these C fiber neurons that are really actually sensitive and become activated for social touch. So when someone is actually gently stroking your skin, these particular neurons start to fire and give feedback to your brain that you're being socially touched. And it looks like we've evolved to have particular receptors that can be picked up using this MNG to look at the effects of social touch. So this is another new way of measuring social neuroscience, new social neuroscience method here, looking at peripheral nerves. All right, so our next class of measures are called hormone measures, and they basically measure endocrinological function. And we actually do cover hormones in greater detail in Lecture 5. But just very briefly right now, I'll just mention that we do have different kinds of hormones that are released by these different glands in the body. Some of them are known as protein or peptide hormones. These are basically made up of chains of amino acids, and they often work via second messenger effects. That is, they cause another gland to release a hormone. You'll see that later on in Lecture 5. We also have these steroid hormones that are derived from cholesterol. And an example of a steroid hormone would be cortisol. And cortisol is, of course, released during times of stress. And then we have like sex hormones. And these are a special form of steroids. And these would include things like androgens, like testosterone. There's also estrogens, like estradiol, and progesterone. So these are all different kinds of hormones that we might be interested in for social behavior. In terms of, does testosterone increase aggression? Do estrogens have something to do with bonding? That kind of thing. So we will be covering like this, like I said, later in the course. But I today wanted just to talk about how do we actually measure these hormones? How do we get them in the participant? So we have 4 different ways that we can measure hormones. One way is to take blood samples, and then once you have the blood sample, you would go to a lab and assay the blood sample for a particular hormone that you might be interested in. The advantage of doing this is that the samples can be then collected throughout the day, and you can measure immediate changes. So like within a short period of time of some stressor or some social stimulus, you could go ahead and ask for a blood sample and then have that sent off to be assayed. But the big disadvantage, as you can imagine, is it is venipuncture. That means that you do have to stick a needle in a person's vein to measure their, pull out some blood. And this can be difficult then to do, just because people are not very happy about having needles stuck into them. It's also fairly difficult to assess concentration changes over time. So you don't really get a really good measure of how much the concentration might be changing of the hormone over time. Saliva is another commonly used measure to look at hormones, a method, and the advantage is that it's not invasive. It doesn't involve you sticking a needle in someone's skin. It can be taken frequently. We've actually done this in my lab group. We've actually used saliva to measure hormones, and you just basically ask a person to drool a little bit, spit into a little test tube, and then you can seal up the tube, put it in a freezer, and then eventually get sent off to a lab to be assayed. But the disadvantages are that protein hormones are not found in saliva, so if you're particularly interested in those, you won't be able to see them. Enzymes and bacteria in the saliva can sometimes change the structure of steroids, and if you have a little bit of blood in the mouth, for example, like let's say you had a bloody gum or some problem with a tooth, that little bit of blood can also give you false high values because you'll be getting basically like a blood test from that person when the assay was looking for saliva. A third type of hormone way is to measure it through urine. The advantages are that you can actually you can look at accumulation of the hormone over time, like you could wait after over an hour to see how much of the hormone has been released. You can look at disease-related alterations in hormone metabolism this way. But the disadvantage is that many hormones are metabolized by the liver prior to them being excreted into the urine. So there's a lag then also between their peak blood levels of a hormone and what you might see in the urine. So there are definitely some differences that you'll get when you do a urine type of hormone collection. But depending on what your research question is, it might be completely appropriate to use that method. And finally, you can do fecal samples. The advantage is here you can be collected from organisms where the close contact isn't possible. For example, maybe you're testing wild monkeys, and you can look at the accumulation over time of hormones in their poo. I remember a long time ago, I knew a woman who would spend time in jungles in Brazil where she would have a small section of the jungle that she was looking at some particular monkey, and she would have to watch these monkeys very carefully and find out where they were pooing, and then she would go and grab some of their samples and then take the samples and get them sent off to be assayed. And what she was specifically looking at was how the hierarchy of the monkeys in this particular troop might be reflected in the hormone levels in the fecal material that she was collecting. The disadvantage is that many hormones are already metabolized by the time they make it into the feces. There's no protein hormones or neuropeptides that can be measured in the feces, so you're limited in which ones you could actually look at in fecal material. But as I said, we'll come back to hormones in lecture number 5. Now, the next class is going to seem sort of strange to have in here for social neuroscience, but believe me, these are very important measures to have in any social neuroscience study, and that would be just behavior and any self-reports, having people make ratings, having them report whether or not they saw the stimulus, what they think about how attractive someone is. So there's a lot of measures, of course, using behavior. I would also throw in here reaction times are going to be important, so how quickly you respond to a stimulus. And you can also throw in eye tracking. So eye tracking is very common these days, where you can have somebody wear like a little set of goggles or have them look through something where they're now watching like those boxes there. Maybe the boxes light up in different colors at different times, and you can actually watch how the their eyes move around on that screen by measuring a reflection off their eyeball as they're looking at this. So it's a really good way of looking measuring exactly what it is that a person's looking at. And I would consider that a behavioral measure, because you might, for instance, want to know if they look at this part of the picture, maybe they have a particular physiological response that's different from when they look over at this part of the picture. So eye tracking has become quite important in social neuroscience. I'll just mention, by the way, that self-report measures themselves, you've probably learned a lot about those in other psychology courses, and all of the stuff that you know about how to take a good self-report measure equally applies in social neuroscience. So we do appreciate well-validated self-report measures in social neuroscience. Behavioral genetics is our next category of methods. And we're going to be talking about genetics a lot in lectures 11 and 12. So again, I'll just give you a quick sample of some of the methods that would be used in behavioral genetics that are important to keep in mind. So in behavioral genetics in particular, there's been a lot of work done with twin and family studies where you go ahead and look at monozygotic versus dizygotic twins and whether they're reared apart or they're adopted out, that kind of thing. So twin and family studies have been, for many, many decades now, have been a really great source for us to understand a lot about genetics of different features in a person, their traits. Now you might remember that these features or traits that I'm talking about would be called phenotypes. So a phenotype is a characteristic, like a behavior, a trait, something that we can see or measure about the person. Even your height is a phenotype. And then the genotype would be what the genetics are that allow you to have that particular phenotype. And so twin and family studies have been done for, like I said, decades to look at that relationship between genotypes and phenotypes. Now, the second kind of class of things here, and I'm throwing a lot in here, is just genotyping, where you actually look at the specific genes. And this is these days commonly done through PCR tests, which means that you can take a sample from inside your cheek or some part of your skin. It doesn't really matter. Someplace that has DNA in it, some DNA tissue. And using this PCR, this polymerase chain reaction method, you can make millions to billions of copies of a specific DNA sample. So that there's enough there for the scientists to then study that sample in detail. And the kind of things they do once they have enough of a DNA sample to work with, they could, for instance, look at what are called candidate genes. And a candidate gene approach means that you have some a priori theory about a gene having a particular phenotype. So for example, maybe someone along the way discovered that this particular sequence of genes is involved in serotonin. And serotonin is an important neurotransmitter. And so what you might do then is try to hypothesize that other behavioral traits that involve serotonin, maybe like depression, whatever it is that you think serotonin might be involved with, you would then try to see if that particular gene is then related to that other phenotype, like depression. So that's the way candidate genes have been used in the past. The problem is that these studies of candidate genes are often difficult to replicate. So somebody else comes along and tries to do this, they don't find it. And one of the problems is that a lot of the candidate gene studies were done with small samples, and so they may have just capitalized on chance, like a false positive was found in the relationship between whatever the gene was that they were looking at and this other phenotype that they thought might be related to it. So the candidate gene approach is sort of fallen out of favor these days, although I'm sure there's plenty of good research that's done with it. Then there's these genome-wide association tests, or GWAS. And what you do in a genome-wide association study is you look at the relationship of millions of different polymorphisms. And a polymorphism is just like a certain thread or a certain part of the genome that you're interested in, and you try to look at how all these different polymorphisms are related to particular phenotypes. Now, these are not usually done theoretically in advance. What you do is you kind of are just using the fact that you're looking at millions of these polymorphisms and seeing how they might be related to all these other possible phenotypes that you're interested in. And voila, you might find a few of them are particularly strongly related to that particular phenotype. And in that way, many behavioral traits have been found to have some of these relationships, but they tend to be polygenic, which means that it looks like the relationships between genetics and some of these behavioral traits actually involve many small genetic effects. So like it's kind of like the sum equivalent of a bunch of small little pieces of this actually might be related to that particular phenotype. So we haven't found just from chance this one particular gene is always related to this social behavior. It's more like there are multiple spots in the genome that might be associated with that particular social behavior as a phenotype. Finally, we have this thing called a SNP heritability, or an SNP. A SNP is, the way you think about a SNP is, you might remember that DNA is made up of these nucleotides, and A, G, T, and C, but perhaps maybe the G nucleotide is in a particular part of the genome in most individuals, but in a minority of them, maybe this position G is actually occupied by an A. And so what that would mean then is you could possibly take all of the people who have the A in that one particular location compared to all the other people who have a G at that spot in the genome, and try to see if there's a difference then between those two groups on whatever the phenotype is. So that G or the A, that one single allele that makes a difference between that particular part of the genome, could be then tested on a large number of unrelated people to see if you can find an association, like a correlation between whether or not you have a G or an A is related to a particular phenotype. And so that's how SNP heritability works. Now, in all of these genotyping studies, and even in the twin and family studies, what they've been trying to do is establish what's called the heritability for a particular phenotype. So this is a statistic that estimates the degree of variation in that phenotype trait in a population that's due to genetic variation between the individuals in that population. So it's a way for us to kind of say how much of this phenotypic trait seems to be due to the same amount of genetic variation that you see between individuals in that population. So heritability is a very common sort of outcome of this behavioral genetics work, is trying to find the heritability of different kinds of characteristics. And we'll talk about those more in those future lectures. Let me just point out, though, there's sort of this quest, for heritability in the case of missing heritability, that if you kind of look at the right there, those are those twin and family studies. And those what they do is they tend to overestimate heritability. So you can see heritability estimates from twin studies might be too high because the genes and environments are correlated in ways that are difficult to measure and account for. And the other idea is that identical twins they might be actually quite different from fraternal twins in the way they get treated. Over on the left, you can see that heritability estimates from measured DNA studies, from these genotype studies I was telling you about, might be too low. And it could be that DNA studies don't have enough people to reliably estimate the small effects of genes. It could be that DNA studies don't measure every genetic variant, and unmeasured variants might have even bigger effects. So this is kind of the problem when you look at these different methods, is how they're doing in terms of really giving you an estimate of heritability. But as I said, we'll come back to this in a future lecture. All right, for the last part of this lecture, I want to go ahead and talk about ways that we study the brain. And this is particularly relevant to social cognitive neuroscience that focuses a lot just on brain activity, that branch of social neuroscience. And the three general ways that we can talk about how we study the brain are with electrophysiology, imaging methods, and lesion methods. Now, for electrophysiological methods, this means that you're going to be doing electrical recording of neural activity. And so one of the oldest ways that you can do, and they've been done this with non-human animals as well as with humans, is to do single cell recording, which sounds like microneurography, but it's a little bit more invasive because it involves drilling a hole there in the skull and then sticking an electrode into the brain and measuring brain activity directly by looking right in there in the cells. And you can imagine this is quite invasive, and they're not going to do this normally on a human unless perhaps the human's already having a surgery for some other reason, like removing a tumor. So this isn't very commonly done, but this is probably, the sort of a gold standard way of measuring neural activity, because you're right in there looking at a bed of neurons and measuring activity that way. So something that's more non-invasive, and instead of measuring it that way, would be to use EEG and ERP recordings. Now, EEG and ERP are basically the same thing. An ERP is actually an EEG in the sense that an EEG is what we call the electroencephalogram, and that's measuring electrical activity that's coming from your head, from the neurons firing there. An ERP is an event-related potential. That's what ERP stands for, and you record ERPs using EEG. Okay, so it's just a type of, like a subtype of EEG that you hear about called ERPs. And for both methods, for using EEG, you're going to use something like this, like a cap with a huge array of electrodes that are known at specific sites. You've measured everything about them exactly in what's called the 10-20 system. You know exactly the location of each of those electrodes that are measuring electrical activity at the scalp. If you recorded a person at an EEG, what you would see is something like this. It can be done on a computer terminal here, and you can see these are all different sites, like the O is from the occipital lobe, T is from the temporal lobe. Odd and even refers to whether you're talking about the left side of the head or the right side of the head. And you can see that just normally when a person's awake, you get lots of different kinds of waveforms. Here you get some slower ones, you get some faster EEGs. And what those EEG waves are do matter in terms of different kinds of activity. We'll talk about those later when they come up. But ERPs are done towards a specific stimulus. So what you see here is a person might be looking at a stimulus up there and they have to make a button press. And so maybe they press the button, and what's going on is like right here at the 0 point, they don't have any activity yet, but within 200 milliseconds, you can see there's already been a change here in the ERP. And finally, we get about the time they make a button press, at about 300 or 400 milliseconds, you can see we get this really big waveform going down. Now, what you see there is actually the activity over many, many trials. So what you would do is you would record that EEG like you saw on the previous slide, and you would do it for maybe 50, 60 times, keep presenting the stimulus you see there up on the left. And then what you do is you average over all of the stimuli, this little section that's after each stimulus that's being presented, because you can see each one by itself down there on the bottom is actually not perfect. It's not showing you the same waveform each time. But if you average across all the same stimuli, you eventually get something like you see in the middle there, a very characteristic stereotype type of ERP form. It's got labeled components like P1, N1, P2, N2, P3. These are all different components that all happen within 500 milliseconds of a stimulus being presented to it. And the amplitude and where how far away they are from the middle of the line, how quickly they change, all of those are characteristics that you could measure about ERPs that may have some particular important function. And then you can also look at how the ERP is collected at different parts of the head, and you might see that there's some lateralized effects of the ERP, or there's something more in the occipital lobe versus the front. And you can even make little contour maps like you see here down in B in the left that kind of really helps you figure out what part of the brain might be more active in that particular task. And just off the top of my head, I can think of a couple studies that used ERPs in social neuroscience. One looked at, for instance, black versus white faces, or male versus female faces, to see if you saw ERPs that were different for people in your in-group, or different because of gender. Does like gender happen before you look at race? Those kinds of questions can be looked at with ERPs. All right, and then we have MEG, or magnetoencephalography. Magnetoencephalography. Oh, geez, that's a hard one to say. And what you do here is you sit in this kind of very strange looking device, and you measure magnetic fields, and they require at least 50,000 neurons being activated to get such a magnetic field. And then you look at these magnetic fields that are generated by all the electrical activity of your neurons. So when you have 50,000 neurons all firing, and they have like these electrical impulses that are going down their axons, they can actually create a magnetic field around them based on how active they are. And then that magnetic field is constantly changing across your scalp or across your brain because of all the different neurons that are firing. And you can use this device to measure those magnetic fields. And it gives you really good spatial resolution that's better than an EEG to kind of see what parts of the brain are active. These MEGs are very expensive. They don't require anything to be injected or anything else. They're actually quite safe. So the person just sits there quite still, and you present stimuli to them while you measure their magnetic fields that are being produced naturally from your brain activity. So those represent all of our electrophysiological methods. Now we're going to move on to imaging methods, where we can take pictures of a person's brain. And we have lots of choices here. You could just do photography. You could take pictures of a brain after an autopsy's been formed, or you have a cadaver or something, and you could look at all the brain slices to look at the brain. We don't typically do that in social neuroscience, but we can look at other kinds of measures and look at structure versus function with them. These first ones are all structural measures. So like there's the CT scan, or the computerized tomography scan. It looks like this, and it's basically an application of X-rays. So there's radiation involved, and you wouldn't want to do this very often with your participants. It's mainly used in clinical settings to diagnose tumors and hemorrhaging. And one of the problems is you can't really distinguish white from gray matter very well with this particular method, but you could get an idea of the sizes of ventricles, and you certainly would be able to see if there was a tumor in the brain. So it's a way of like using X-rays to image the brain. And then we have MRI, or magnetic resonance imaging, and there's lots of different ways. You can basically do an MRI of any part of your body. So you can see in the picture here, they're doing an MRI of this person's lower waist. So there's lots of places you can look at with an MRI. Here's an MRI that's taken of the head, and it's just looking at the structure. So it's a structural image of the person's head, and allows us to look at both white and gray matter tracks. You can really clearly make out the different parts of the brain using this particular method. The person has to be sitting very still during this, and you're going to get some nice images here. You could measure, for instance, how big the cerebellum is, or how big their amygdala is. There's lots of things you could do, and you can actually see these images in 3D, so you can actually look at from different sides and so on of what the MRI is. Then we have functional imaging measures of brain activity. And these don't actually measure the neurons directly. What they do is they measure the consequence of the neural activity. And some of our most common ones would be PET, positron emission tomography. And this is what PET images look like. Now, what happens in a PET is you actually have to use radioisotopes. So you have to have some place that's got a cyclotron that's going to create these radioisotopes. There are not that many around. There might be one in Brisbane, I don't know. But what you would do is use the cyclotron to create the radioisotope. And then the radioisotope only has a certain length of time where it's going to be any good. It's got this half-life decay on it. And then you would insert that radioisotope into the person while they're performing a task. So what you do is you get this radioisotope and you get it all ready to go. And then you're going to give it to the participant through like an injection. And as they start to do their task, the radioisotope is being taken up into the blood and up into the brain. And it's going to allow us then to look at what parts of the brain are active while the person's doing this task, while the isotope is there in their system. And so what you would do is you'd maybe have them do one kind of task, then have them do another kind of task, and look at each time after you've done that, go ahead and have them take a picture, put them in the PET scanner, and then you could see how the brain has taken up that radioactive isotope. And it's basically letting you get an idea of what part of the brain is functioning the most, what's being used the most by that radioactive isotope, the way the blood is flowing. So you can see here are some PET images of a person at rest. Here's where they're being presented visual stimuli, and you can see near the back there in the occipital lobe, it's more active. There's more of the radioisotope being put there. So as you can imagine, PET scans are kind of expensive. They're very difficult. They do allow us to do certain kinds of things that you couldn't use, for instance, with an MRI. But the more common thing these days is to use a functional MRI. And it's like that MRI that I showed you before, except that we now, on top of the functional MRI, are going to be looking at what's called the bold signal. So we'd have our participant lying there in the fMRI scanner. We have actually a couple of these at the University of Queensland now. And what you can do is have a person look at a computer screen, and you can project things to them. They could watch videos. They could press buttons on a response button, but they have to stay very still. Talking or moving their head very much will actually mess up the images that you take. So a person in an MRI has to stay very still while you're imaging their brain. Now, under that, what are you doing? You're actually looking at what's called the bold signal, and you're looking at basically how much oxygen flow in the blood is changing based on what part of the brain is more active. And the idea is that if a particular part of your brain is more active, it's going to need more of this oxyhemoglobin, more oxygen brought to that part of the brain. And so you can actually check and look at how the capillary beds around particular brain regions are flowing with their oxyhemoglobin. And so the neural activity is going to increase blood flow, which increases oxyhemoglobin. And then you're going to be able to see this using an MR signal. And it's going to allow you then to get an image of the brain. So the key concept here is what's this thing called a bold response. And you can see the bold response, by the way, is sort of slow on the scale of things. It can take a couple seconds there before you actually get the peak bold response. So what you might want to do is have a person do a task for a little bit, pause, do another task, pause, do another task, pause, so that you don't get the bold response too much overlapping with the next bold response. Although there are now mathematical ways to go ahead and treat those bolds coming right after one another. The original fMRI studies were done back in the 90s, and here's this early study from 1992 where they used a flickering checkboard that was on for 60 seconds, then off, then on and off. And you could look at the brain activity using the MRI, and they found that with the fMRI, you could actually look at occipital activity and see when it was off versus on, when it was off versus on. And it was very exciting to kind of show this relationship between a psychological variable, like perceiving a checkerboard being on or off, and actually seeing the direct brain activity in the image. You, of course, can also measure brain activity lots of different ways, both horizontally, sagittally, getting a whole 3D perspective of the brain activity, and therefore you could look at any of these structures in the brain that you really want to do. Now you might think, great, in fact, 20 years ago, it was all the rage to have major psychology and neuroscience programs all around the world at their universities that they each would get an fMRI scanner because they thought this is the way to go. It's going to be the future for the 21st century. Everybody's going to want to have an fMRI scanner that they can share and use for their participants. Now, there's still excellent work being done with fMRI. I'm not saying it's gone away at all, but I don't think it's ever taken off at the same rate that everybody imagined it was going to take off in 20 years ago. Partly it's because it's sort of limiting what you can do in an MRI, particularly when you're talking about social psychology and social behaviors. You can't have a person actually interact too much with somebody. They're kind of lying there still and so on. And also, the other problem with MRI is it's expensive. You actually get charged every time you use an MRI scanner to pay for the overall maintenance of that gigantic scanner, which is a gigantic magnet, basically. And therefore, for instance, at UQ, when we want to do an MRI study, I think the last time I checked, it's around $600 an hour if I want to do an MRI scan. So that's like one participant that I might be testing in my study. I'm going to have to pay the university $600 to use the MRI scanner. And if I'm going to go ahead and have 10, I've now got $6,000. If I want to do 100, you can do the math. So it becomes really expensive to do an MRI study with a lot of participants. And so I think that's one of the reasons why it's not as popular as people thought it would have been 20 years ago. But like I said, there's still some really great research being done, even at UQ with all of our scanners and lots of researchers who get really large grants to pay for these MRI studies. So you will hear about fMRI from time to time in the coming weeks, in the coming lectures. But let me talk to you a little bit about how not to do an imaging experiment. This comes from an old course or slides or a website called fMRI for Dummies. And what's saying is, this is sort of funny. What it's trying to say is, here's not to do an imaging study. Like, number one, ask a stupid question. Like, I wonder what lights up for daydreaming versus rest. That was kind of what people did a lot when they first got their fMRI scans. They just said, well, let's see what lights up when a person does one kind of task versus another. Compare poorly defined conditions that differ in many respects. Like, I'm going to see what it looks like when people are watching happy movies versus sad movies. And sure, that might sound like a real clear thing, but there's actually probably a lot of differences between happy and sad movies, and how do you know which ones are actually affecting the brain activity? You could use a paradigm from another technique like cognitive psychology without optimizing of the timing for the fMRI. And so this is making that point about the bold signal. You remember how it's kind of slow, how the blood flow changes? And so if you're doing some kind of task from cognitive psychology, for instance, it might not be appropriate to do it with the same sort of timing in an MRI. Never look at the raw data, the time courses, or the individual data. Just plunk it all into one big stat model and look at what comes out. So this is the problem that a lot of people get tons and tons of data from an fMRI study, and they could just milk it all they want, run all sorts of analyses until they find something significant. There's just so much data that you get from an MRI study, and that's sort of making a joke about that. Publish a long list of activated foci in every possible comparison. So this is the idea that you have all these different points in the brain that you could look at. And what you do is in your results section of your article, just say, here are all the different foci, and here's every single thing that came out significant, and who knows what to make of that. Double dip and don't use any statistical corrections. There's a famous article that was published by Vu et al. In 2009 called Voodoo Correlations in fMRI Studies. And the idea here is that a lot of people actually do lots of multiple statistical tests in their fMRI studies and weren't correcting for the fact that they could just find things by chance. Number 7, write a long discussion on why your task activates the subcortico-occipito-parieto-temporal frontal network. That's kind of a funny one because that's basically the entire cortex. And finally, bonus points, if there are areas that you don't want to see, just color them gray so they don't show up well. And this has actually happened. There's a study, for instance, that down in the lower right corner, you can see that there was a part of the brain that was also active in this task. And what the researchers did is just grade it out because they didn't actually have any theory about it and didn't want the readers to be distracted by that part of the brain that was also lighting up. So they just manipulated their image. Not a good thing to do, but that's just a point here that this slide is trying to make. All right. Now, the other thing to think about with fMRI is I just want to point out that there's some sort of subtraction logic to fMRI studies. That's one of the problems with fMRI. The idea actually of subtraction logic goes back to Donders, a Dutch physiologist who was doing reaction time experiments. And his idea was if you measure the time for a process to occur by comparing two reaction times, one which has whatever the component is that you're interested plus the new component that you're interested in. So for instance, let's say T1, the task 1 is you're supposed to hit a button when you see a light. Task 2 is you hit a button when the light is green but not red. And task 3 is to hit the left button when the light is green and the right button when the light is red. Well, if you take T2, the amount of time it takes you to hit the button when the light is green but not red, subtract the amount of time it just takes you to hit a button when you see a light, that helps you then measure the amount of time it takes to make a discrimination between light color. Whereas T3 minus T2, both of those involve making decisions. And so you can see here, hit the left button when the light is green, the right button when the light is red, versus hit a button when the light is green but not red. You can take the difference there to find out the time that it takes to make a decision. So the idea of using subtraction logic is you go ahead and take two different tasks and subtract them out, and then whatever's left over is what you think is the new process. So it has this assumption of pure insertion that you can insert a component process into a task without disrupting the other components, which turns out to be not the best thing to think. But here we go. You can see here's a simple experiment in an fMRI study. You can maybe show people intact objects versus scrambled objects. And so we have times when scrambled objects are shown, time when intact objects are shown. So T1 is viewing intact objects. Task 2 is viewing scrambled objects. T3 is when you're resting. You're just looking at a fixation point. And so again, you can measure T1 minus T2, and you can find out what parts of the brain are active that are just distinct to when you're viewing intact objects versus scrambled. T2 minus T3 tells you just about what the difference is between looking at stimuli versus being at rest. So that lets you see, and those colors that you see there are subtractions. So anytime you see an fMRI scan with different colors on the brain, that's because they're comparing two conditions and saying, if I take this one condition that's really close, but just a little bit different from the other condition and subtract them, this is the color, this is the amount of difference that I see in brain activity. And then they usually use like a bright red to show you there's a lot of difference between the two conditions. A yellow means there's only a little bit of difference between the two conditions. And so the point is that you could see perhaps what part of the occipital lobe is more involved when you're looking at object processing versus visual activation. So the problem then we have when we're thinking about these fMRI studies is that we can really be swayed by the fact that they're so expensive and they involve all this fancy equipment and these really pretty pictures of the brain. But I want to warn you that whatever you've learned about other research designs in psychology and neuroscience, they apply to fMRI and other neuroscience research. So what makes a good study, like good sample sizes, whether or not they have adequate control conditions and so on, you should always be thinking about the good ideas of research designs. As you read chapters in the book or you read any articles, be sure you ask questions about the internal and the external validity of the research. So as you look at different people's research in neuroscience, you should always be thinking about the same things you've learned in other courses about internal and external validity. And don't worry in our course too much about these technical details. So if you pick up an fMRI study, you'll see they talk about things like the type of T1 contrast, the rate of acquisition. That kind of stuff isn't really necessary for social neuroscience, this course. But if you go on in neuroscience and you want to do much more about it, and you particularly want to do imaging, there are actually courses that you can take just focused on fMRI, for example, that will teach you much more about the physics behind it, how that scanner really operates, and so on. But we're not going to be caring that much about those details here for this particular course. Now, the last way that we can study the brain is to look at lesions. And lesions are basically damage to the brain. And so if you can think about ways of acquiring brain damage, so there's people out there who have brain damage. And how would this have happened? Well, it could be from neurosurgery. Maybe someone gave them a lobotomy because they were really having problems, or maybe they were having epilepsy and there was the epilepsy was so severe that there was some neurosurgery done. It could be that they have a stroke or some other cerebral event that's caused them to have some brain damage because of the stroke. There could be traumatic head injuries or TBIs, where they maybe you got tackled really hard or were in a car accident. Now they have this traumatic brain injury. People have tumors, and maybe after the tumor's been removed, there's still some damage left over from the tumor. There are viral infections where the infection can damage part of the brain. And finally, we also have neurodegenerative disorders. So all of these things, neurosurgery, strokes, TBIs, tumors, viral infections, and neurodegenerative disorders like, and by the way, I mean things like Alzheimer's. These different kinds of brain damage can affect different parts of the brain. And now you have patients like this that you maybe would compare to patients who don't have these particular problems. So in neuropsychology in particular, you see a field that has a lot of these kinds of case studies or small numbers of patients. Neuropsychology research usually has a fairly small sample because it's hard to find these kinds of people in any great numbers. And then what you try to do when you are a neuropsychologist doing research and related to neuroscience here is that you try to look for single dissociations. So this is a situation in which your patient is impaired on one task, but they're relatively spared on another similar related task. And so then you could say, wow, it looks like this particular part of the brain is involved in this task, but it's not involved in this other task. The other kind of dissociation you could try to find is something called a double dissociation, where you get two single dissociations that have a complementary profile of ability. So they might not be very good at task A, but they do task B really well. But then you might find that another person has damage to part of the area where they don't do task B very well, but they do task A very well. And now that starts to tell us between those two patients that these two particular brain areas might have complementary abilities or parts of the brain that are involved in that. What the researcher is usually trying to rule out in neuropsychology is what's called task resource artifact. That is, if two tasks share the same neural or cognitive resource, they kind of have a common third pool of the resource they use, but one of the tasks uses more, then damage to the resource could be what's affecting one task more than the other. So it's not that you've actually located the brain area where this particular process is done. It's more like you've damaged the damage has actually been happened to the common resource that both tasks use, but one of the tasks needs that resource more than the other one. Then there's also this thing called the task demand artifact, where one task is performed and it works better than another because the other task is just performed suboptimally. So it could be that just by a fluke that the task that you had the participant do, the instructions weren't very clear, for example. And so it's not that there's really a brain difference between the brain region here is really involved in task A but not task B. It's more like task B wasn't performed very well because the instructions for this new task were not done very well. So these artifacts are things you're trying to rule out. And if you can find a double dissociation, you feel much more confident that you've ruled out both the task resource artifact and the task demand artifact. Now, I'll give you a real life example of this. This is something that goes back to 1991, Kubeli, who had two patients who it turned out they had a lesion that led them to having trouble writing or spelling vowels, but they had no problem with consonants. So they couldn't actually write down a vowel, or they couldn't speak a vowel, they couldn't spell. When they made spelling errors, they would make spelling errors around vowels. But consonants were just fine when they were doing their words. Okay, so that's a single association. It kind of tells us that if you have a lesion in this part of the brain, it seems to be the part of the brain that functions in helping us deal with vowels. And that's distinct from the way our brain and another part of the brain might just help with consonants. And then interestingly, just a few years later, Kay and Hanley reported in the literature that they had a different patient who made spelling errors only with consonants. So the part of the brain where they had damage, this particular participant, they did fine with vowels, but they were really bad at consonants. And so the combination of these two studies gives us a double dissociation, where we can see that these two different areas that are being affected by where these patients might have had a stroke or a lesion, one part is really a part of the brain that seems to be processing vowels, and the other part of the brain seems to be processing consonants. And we can rule out those two artifacts because they don't seem to have any bearing here when you have a double dissociation. All right, so that's double dissociations that we try to find. They're kind of like the holy grail in doing neuropsychology research. Now, some caveats about these lesion studies. Keep in mind that some of these lesions are better. The best kinds of lesions for research purposes are those that are which the dead tissue has been replaced by cerebral spinal fluid. So when you have strokes or viruses or neurosurgery, it's best to have that kind of lesion where there's a stroke or virus or neurosurgery, because the damaged tissue is gone and it's been replaced by some sort of cerebral spinal fluid. It's much harder when there's actually when a person's had a tumor or a head injury, because the swelling that happens there can render a lot of the surrounding neurons around that area inoperative, even though they aren't destroyed. So the tissue is still there, and the tissue looks like it's not been damaged, but the neurons themselves aren't really functioning because the tumor or the head injury actually caused a lot of damage. And then there's also this problem about lesion studies, the problem of diaskesis, which is that even a very discrete lesion, just in a very small little part, could disrupt functioning of very distant brain regions. So it could be that this lesion is in one part of the brain, but it affects functioning in another part of the brain far away from it. And so you would mistakenly think that this lesion is the place where that functioning of the brain, where this function is located, but it's actually because some tracks, some axons aren't making it to the other part of the brain where the process really does occur. And you wouldn't know this because the diaskesis gives you this misrepresentation about where the function is actually located. Finally, you can imagine that lesions with patients is kind of hard to do. And so there are some alternatives to this. The big one you can use is what I call virtual lesions. And that's to use TMS, or transcranial magnetic stimulation. And what you do is you change the electric current. A change in this little coil that you see there next to the person's head generates a magnetic field. And then that magnetic field, like little bursts, disrupts the neurons that are just below that in the scalp. So right there in the cortex. And so just for a very brief time there, you can disrupt the electrical activity of synaptic potentials as they are moving along the axons, and cause a disruption of the neurons that might be happening there. And that way you could go ahead and be giving a person a task, like let's say you show them positive versus negative pictures, or trustworthy faces versus untrustworthy faces. And if you think that particular part of the brain might be involved, you could go ahead and try to disrupt it a little bit and see if that affects their judgment. The participant won't feel anything. It doesn't harm them. It's really fairly safe to do. I mean, there's not really a problem here. And we actually do TMS at UQ. There's a few people in psychology, for instance, have used TMS quite well. And they use TMS usually in conjunction with an MRI scan, so that you can be really confident that you know exactly where you're putting the TMS coil right over a particular part of the brain. So they kind of match it up using this sort of nice mapping technology between a person's MRI scan and where the TMS goes. Okay, so we've now covered a lot of different kinds of methods in social neuroscience, again, particularly with humans. We've covered peripheral measures, hormone measures, behavior and self-reports, behavioral genetics, and lots of ways to study the brain, including electrophysiology, imaging methods, and lesion methods. And keep in mind that we'll come back and talk about a lot of these in much more detail in coming weeks, including hormones, behavioral genetics, and a lot more about brain activity. And that's the end of today's lecture.